Hello friends, welcome to Dr. Ossai Physiology Academy, D.O.P.A for short. This is the place where we make the learning of physiology easy, exciting, and effective. Thank you for joining me. So today we are going to be continuing with this topic that we started the previous lecture. So this is part 2 of the general structure and function of the kidney. Okay? So, but I would like you to remember that learning about how the body functions, how it works, which is what physiology is all about, can teach you so many lessons, health lessons, life lessons, and even management lessons. Let's get started. All right. So in the previous lecture, we started with the surface structure, we call it gross anatomy, okay, of the kidneys, the different parts. It's really for descriptive purposes, just like you want to tell somebody your house address. If it's properly mapped, location, GPS and all of that, get the exact location, longitude and latitude. So that's basically what gross anatomy is for. But it cannot really tell you so much about why something functions the way it functions. It's for descriptive purposes. But then there's always more than meets the eye. So for you to really understand how structure determines function. Remember we said that structure determines function. You need to go deeper. Okay? And that's what they call it histology. That's at the cellular level. You need to zoom in and see how the cells, because the cell is the structural and functional unit of life. So when you study things closely at the cellular level, you get a lot of insight. So that's what we're going to be doing. So we said that this nephron is the functional unit of the kidney. And we started by saying that it's divided broadly into 2 parts. The renal corpuscle, that is this area, which is made up of the glomerular capillaries and the Bowman's capsule. Then from here down is the renal tubule. So we discussed this first part, the renal corpuscle. And that's where you have the filtration barrier, because broadly speaking also, the kidneys, you can divide their function into 3. One of them is filtration. Another one is reabsorption. And another one is secretion. Okay? So this first function, filtration, the renal corpuscle handles it. So we went deep into the filtration barrier, looking at how it is structured in order to perform adequately this filtration function. We looked at everything, the cells there, the different 3 layers. You can go to the lecture and listen to it, okay? Watch it there. So now the remaining 2 functions of this nephron is by the tubules, reabsorption and secretion. So you ask yourself, what does reabsorption, what does it really mean? Why it has to be re? Why not just absorption? First of all the resources we have in our bodies, everything, the elements, we get them from outside, from the external environment when we eat. All the sodium, the carbon, every element found in your body, they are taken through the mouth, okay, majorly. So when you're taking food, they are absorbed. They are broken down through digestion and they are absorbed into your bloodstream. So this filtration now takes it from your bloodstream. When it filters it out, it just leaves the cells, red blood cells and large molecules, plasma proteins away. But every other thing is filtered. But your body still needs those things. They are not wastes. Okay? So it filters both the wastes and necessary things. Even glucose, as important as glucose is to the body, glucose is filtered too. But the body needs to take it back to the bloodstream. So that's why it's called reabsorption. So anything that goes into the bloodstream, it goes through absorption. So this one, the absorption has happened when you ate food and Glucose entered into your system. Now glucose came out. So it's being read. That's why it's called, but you can still call it absorption. Okay. That's that. Then secretion is about the opposite of reabsorption. But before we go into that, let me just let you know this, that after this, we said this is the afferent arterial, okay, that leads to this glomerular capillaries. Then this is the efferent. So what happens is that this efferent now forms normal capillary. But we know that this one is not really a normal, typical capillary. It does only filtration. But this efferent now leads to another set of capillaries that surrounds the tubules, okay, all the way down. So you see it. So this efferent, these are capillaries, and they are called peritubular capillaries. Again, from the name, you already know what it means. Peri means surrounding. Okay, so just think these capillaries that surround the tubules. So these capillaries are the ones that now perform the normal functions of capillaries. The reabsorption takes place majorly there. Okay, so around all these areas here is normal. You have normal interstitial tissue fluid, interstitial fluid, and all of that. Okay, so reabsorption happens like this. When resources are filtered, as the plasma, they are filtered into this tubule. Because some of them are still needed in the body, and the concentration is usually higher here. So what happens is that they now get transported through this tubule into this interstitial space, okay, to the interstitial fluid. Then from the interstitial fluid, they now get transported again into the capillaries. It now enters the bloodstream goes back to the bloodstream. That's what happens in reabsorption. So from the tubule to the interstitial fluid, and the interstitial fluid back into the capillaries, into the bloodstream. So secretion takes the opposite route. Okay? So now listen very carefully. When blood enters from this afferent arterial into this glomerular capillaries, not everything is filtered. Okay? Not everything is filtered. Just about 20%. We'll talk more about glomerular filtration rate and all of that. So just about 20% is filtered. So 80% enters this efferent arterial and the peritubular capillaries. So what does that tell you? It means that there are still some substances, some wastes that are in that 80% that the body still needs to take away. Okay? So they now have another opportunity to enter into the tubule. So they enter these efferent capillaries, these efferent arterioles and the peritubular capillaries, and then from the capillaries, they enter the interstitial fluid. From the interstitial fluid, they now enter the tubule. Then it moves and is excreted. Okay, so secretion is just the opposite of reabsorption. So those are the two major functions of the tubules. Okay, so we are talking about movement of substances into the capillaries and from capillaries. But how do they happen? Okay, like I said, the histology, the cells, looking at the cells will tell you a lot, because everything happens through the cells. Okay, so it's basic transport mechanisms. There are transport mechanisms by which these things happen. You know, in general physiology, you will learn about diffusion, simple diffusion, facilitated diffusion, primary active transport, secondary active transport. All these processes, they are used in the transport of substances, the reabsorption, and even the secretion. Okay? So details of that will be learned in subsequent lectures. But so, the next thing we need to do now is to now look at the different parts. You know, this is a long tube. So we need to know the names of these various parts. So that when we are talking, you easily understand. And they are unique. Each segment of this tubule, they are unique in their own way. Okay? So we're going to be talking about the characteristics of each segment that help in this reabsorptive function and the secretory function after this break. Welcome back. So now we're going to be talking about the different parts of the nephron so that we understand their peculiarities. Now, after this renal corpuscle, the first part of the renal tubule is called the proximal tubule. Okay? This first part. And is divided into two parts also. You have the convoluted part and the straight part. Okay? So you have the proximal convoluted tubule and the proximal straight tubule. Look at it. This convolution, that means it's talking about that it's bending, it's windy, it's not straight. So convoluted is the opposite of straight. So you see this other part from here to here is still the proximal, but it's straight. Okay? Although some texts will tell you that this is thick descending. They put it as part of this next part, but prefer we name it like this. It's straightforward, more straightforward. Proximal convoluted tubule and the proximal straight tubule here. Convoluted, here, straight. Okay? So the next part is this from here. You see that if you watch very closely, you see that it became thinner. Here it's wider, it became thinner. Here like this, then it became fatter again up to this place. So the next part we have what is known as the loop of Henle. The loop of Henle. That's from here to this place. So the loop of Henle is divided into parts also. So you have the descending part that's from here. This is the descending part. Then this curved part is known as hairpin bend. Okay? Descending, the hairpin bend. That's this sharp curve here. Then you now have the ascending. Okay? So this descending and the hairpin bend, they are thin. If you look at it very closely, both of them are thin up to this point. So the ascending has two parts. The thin ascending, limb or part. Then from here it became thicker. So here we call it the thick ascending limb of the loop of Henle. Alright? Hope you got that. So the next part is now the distal tubule. The distal. So where do we start? Where does the distal tubule start from? You see this red marking here. That's what demarcates the thick ascending from the distal. So from here onwards, it's known as the distal tubule. Okay? Then we have another part. But first of all, the distal tubule is also divided into two parts. You have the early distal tubule, and you have the late distal tubule. Then we'll have the fourth final part. It's called the collecting ducts. Collecting ducts. Alright? Some will initially call it collecting tubules. Because the interesting thing about the collecting duct is that when it reaches the collecting duct, several collecting ducts join to form larger, progressively larger and larger and larger and larger ducts. So initially you can call them collecting tubule so that it will still match this nomenclature. Then when it became larger around this point, collecting ducts. But you can just, for simplification purposes, collecting ducts from after this late distal. Okay? So these are the four major parts. Just get that into your mind. Proximal tubule, convoluted, straight, loop of Henle, descending, ascending. You have the thin part, the whole descending is thin, and the hairpin bend. Then the ascending part, you have the thin and the thick part. So now, what is special about this proximal tubule? The very special thing there is that it is where most reabsorption takes place. And what helps it to do that? Now, it is, you see that it's fat. It's made up of cuboidal, all these things here. A single layer of cuboidal cells. It's called cuboidal cells. Okay? Because of their shape. So this edge, all these places you see here, the edge of everything, the whole tubule, they are cells, single layer of cells. These transport processes that take place, they take place through cells. Okay? So that's where you need to revisit your transport mechanism, transport across cell membranes to help you understand. It's the same thing, the different various forms of transport. So there are two cuboidal cells. But apart from the cuboidals, because other parts still have cuboidal cells, the significant thing here is that these cuboidal cells, they have what is known as microvilli. Microvilli, they look like hairs. Okay? Microvilli. The main thing there is that the microvilli help to increase the surface area for reabsorption. That's why it does most of it. It increases the capacity, the area that reabsorption can take place. So it's also known as brush border cells because of this microvilli. Alright? So about 60 to when water and solutes are filtered, about 60 to 70% of them are reabsorbed just in this proximal tubule because of this glucose, sodium, everything, water, 60 to 70% of them. In fact, glucose, because of how important it is, 100% of glucose is reabsorbed. But generally, 60 to 70% of solutes, 60 to 70% of water is reabsorbed in this proximal tubule, both the convoluted and the straight parts. Alright? So the descending part, you see how thin it is. It's made up of flattened epithelial cells. They are all epithelial cells, okay? Cuboidal epithelial cells. So because it's flat, this whole flat part is made up of flattened epithelial cells. So the unique thing about this loop, it's called a loop because of the shape, is that it majorly helps in concentration of urine. There are special types. We will talk about that briefly before the end of this lecture. There are special nephrons that have very long loop of Henle. They have very significant role to play in concentration of urine. We'll visit that later. So that's just that. Little reabsorption takes place there and some secretion, okay? But the same thing with this thick ascending limb is tuboidal cells, okay? Both processes also will take place there. But we'll talk about that much later. So another significant thing to mention is the fact that from here, you can see how the shape is. It came back. You see, this thick ascending limb came back to meet this glomerulus here. You see, that's why it's labeled red. It actually touches it. It forms a unique structure. This is the dividing point between the thick ascending limb and the distal. So this point here is known as the macula densa. There are cells here. They are known as macula densa. They have special functions. We'll talk about that. Okay? So, this macula densa joins with the glomerular capillaries and some other special types of cells here to form what is known as the juxtaglomerular apparatus. Let's write it here. Juxtaglomerular apparatus. Okay, let's go to the end and we'll visit juxtaglomerular apparatus specially. There's something one or two facts we need to learn from there. Okay, so after that, we go over to the collecting ducts. And one thing you should take home is that the thick ascending limb is very similar with the early part of the distal tubule. Okay? They have functional similarities, similar kind of cells, thick ascending limb and the early part. So then the late distal tubule has similarities with this collecting duct. Alright? So the early distal tubule has similarity with the thick ascending limb, and the late distal tubule has very close similarities with the collecting duct. And what are those similarities? The late distal tubule has intercalated cells. Okay? While the collecting ducts, they have, in addition to intercalated cells, they have what is known as principal cells. So those cells, they have functions related to the secretion of hydrogen ions, acid, and all of that. More of that later. Alright? So that is generally the different parts of the tubule, the renal tubule. Okay? So these collecting ducts, they progressively become larger. The collecting ducts, they progressively become larger and larger till you have just 250 of them that now empty into the renal pelvis, and from the renal pelvis to the ureter. Okay, so about 4,000 nephrons empty into the collecting ducts, making 250 large pelvis. So you see the arrangement. So that's that. So next, before we round up, we'll deal with this juxtaglomerular apparatus. It's very important for us to know. Okay? Right. So the juxtaglomerular Juxtaglomerular apparatus. Okay? So this juxtaglomerular apparatus is made up of 3 parts. Number 1 is this macula densa that we have mentioned, as special cells. Then 2, we have juxtaglomerular cells. Okay? Straightforward, juxtaglomerular cells. Then 3, we now have what is known as extraglomerular mesangial cells. Look at it, mesangial. Extraglomerular mesangial cells. They have a second name. They are also known as laces. Laces cells. So these 3 together is known as the juxtaglomerular apparatus. So what do? Now this, we talked about secretion of renin at the previous lecture, the general functions of the kidney in regulating blood pressure. These are the cells that perform that function. They secrete renin. Okay? So they play that role in the regulation of blood pressure. That's where then the macula densa. This macula densa, they are osmoreceptor cells. What do you mean by osmo? It means that they can sense the concentration of this filtrate. When the filtrate gets here, they can sense the concentration. And if there needs to be a change, they sense it and they pass that information to this place. So generally they help to regulate this filtration process here, glomerular filtration. And these extraglomerular mesangial cells, they are contractile cells. That means they have the ability to contract. We can also call them pericytes. Another name is known as pericytes. Okay? So they can contract, and their contraction can help change the diameter, the diameter of this glomerular capillaries, which also helps to regulate filtration, glomerular filtration. So generally, these are the function of the different cells together. That's why it's called an apparatus or juxtaglomerular complex. Okay? So that's basically what you need to know about the tubule, the renal corpuscle and the renal tubule, different parts. So what lessons can we learn from here? Very simple. You can see division of labor. Division of labor. I'm sure you've heard of division of labor before. So for you to be effective, you need to divide responsibilities. Each part has their own special responsibilities. Not that you just have a nephron and every part performs the same function, reabsorption and all of that. Different parts perform a different function. And that reminds me of the different types of nephrons. You have the cortical nephron. The kidneys has cortex and medulla. So the cortical nephrons, they are shorter. This renal corpuscle starts from the outer cortex and ends at the junction between the cortex and the medulla. But the next type of nephron is the juxtamedullary. They start from that junction of the cortex, and this loop of Henle is very long, goes deep down into the inner medulla, very long. And their special role is in the concentration of urine. They help in the concentration of it. The juxtamedullary, they're about 15%. Some texts will say 20%. So 15 to 20% juxtamedullary, then 80 to 85% of nephrons are cortical nephrons. Okay? So you see the demarcation, structure, division of labor, function all leading to effectiveness. So if you want to be effective in any system, different parts, different people need to have different responsibilities broken down. Not everybody doing the same. When everybody is doing the same thing, you can't get the best. So there's division of labor and specialization in the whole system, the functional system and unit of the kidney. So in the next lecture, I'm going to go deep down into glomerular filtration. A lot of things we need to understand, the things that regulate the filtration. And also we'll talk about the specific substances that are reabsorbed, filtered, and all of that from the next lecture. So if you want to do further reading, I have some books that I've written, even on renal physiology and other parts of physiology. Just check the description box. You'll see the link to Okada books. You can download it just for a token for further reading to get a better grasp of these things. All right? See you in the next lecture. Thank you.